much for putting this wonderful symposium together. It's really been a pleasure to hear everyone. So let's see if I can. There you go. So what I'd like to talk to you about today is our path on understanding disease circuitry at single cell resolution and how we can use that to actually start reversing some of the disease phenotypes that we're discovering. The basis for all of this work starts with a huge revolution in genetic studies that has revealed 120,000 loci across the genome associated with diverse disorders, enabling us to get an unbiased view to disease mechanisms, discover new target genes, new therapeutics, and ultimately enable precision medicine and even personalized medicine. The challenge with these genome-wide association studies is that if you open up the hood and you look at what are the actual genetic variants underlying these disorders, what you see is that in the vast majority of cases, those variants actually do not perturb the proteins directly. Here's one example from body mass index, a measure of obesity, and the FTO locus, where 89 common variants in that region of association mostly fall in the first intron of the FTO gene, but do not perturb the FTO protein directly. So the question is, how do we even know whether FTO is the target gene? What is the target gene? In this particular case, the true target genes are actually millions of nucleotides away, and FTO is just a red herring that apparently doesn't seem to have a role directly in obesity. What is the causal variant among all of those genetic variants that are co-inherited with a disease? If we don't know the causal variant, how do we even figure out what cell type these variants are acting in to basically know how to even build our therapeutic hypothesis and modulation? And how do we go about discovering the relevant pathways and the specific mechanism? So that's where a lot of the work that our lab tries to do falls in. We basically try to systematically integrate genetic variants, both common and rare, both non-coding and coding, into circuitry and pathways. How do we build that circuitry? By systematically profiling RNA and epigenomic information systematically in both healthy and disease samples to basically understand what are the control regions that underlie disease and how are these control regions linked to both target genes and upstream regulators that control them. We generate massive amounts of information, which we then integrate computationally to predict driver genes, regions, and cell types. And then we systematically validate these predictions, both in human cells and in mouse models, and of course, disseminate the results and complete the cycle all over again. In the end, what this gives us is the circuitry for every one of these hundreds of thousands of disease regions. We basically have the upstream regulators controlling specific enhancers, the cell types where these enhancers become active, which SNPs perturb which enhancers, and what are the target genes downstream. So with every region of association, we can now go and figure out the complete circuitry of this region and understand mechanisms underlying this. Going back to the first slide, what this gave us for this FTO locus is a circuitry that doesn't even involve the FTO gene. Instead, there's a single nucleotide variant somewhere in that intron of this gene, which basically disrupts binding of ARI5B that can no longer repress this super enhancer in this locus, which acts in adipocytes to repress IRX3 and RX5. So without that repression, when the motif is lost, you basically have de-repression of IRX3 and RX5 that are sitting 600,000 nucleotides and 1.2 million nucleotides away from the disease variant, which then shift from thermogenesis in normal individuals, in lean individuals, to lipogenesis in obese individuals, thereby resulting in the storage of all of the excess calories in our diet as fat that accumulates rather than burning it off as heat. And the advantage of figuring out circuitry is that we can then manipulate circuitry. We can go in and turn on or turn off the upstream regulators or the downstream target genes. We can even go with CRISPR-Cas9 genome editing and reverse that single nucleotide variant to establish causality at the nucleotide level from C to T and T to C. And in all of these cases, observe both the cellular and the organismal phenotypes of these perturbations. And what we find is really striking. It basically shows that we can actually shift between lean and obese phenotypes like a switch. We can basically restore cellular thermogenesis at the cellular level with a single nucleotide perturbation out of 3.2 billion nucleotides in the human genome. Changing that single letter basically restores the cellular phenotype with a 7-fold increase in thermogenesis. And in the mice, you basically see the organismal phenotypes of actually downregulating ARIX3, one of these genes that is de-repressed in obesity, downregulating, and it showed that mice lose 50% of their body weight, and they are unable to gain weight in a high-fat diet. When normal mice gain weight, these mice are unable to gain weight. They simply burn their calories in their sleep. So what I'm going to describe today is how do we now move this to thousands of loci systematically. So first of all, how do we build reference epigenomes to predict disease-relevant tissues and states? How do we combine genetic, epigenetic, and transcriptional variation in disease? How we dissect epigenomic and transcriptional circuitry at the single cell level? And how do we understand cell type specific genetic effects across thousands of loci across the genome? How do we then move from one phenotype at a time to multiple phenotypes, and from one tissue to multiple tissues, and their convergence across multiple disorders? And then lastly, how do we systematically manipulate these elements in high throughput? So first of all, to understand the epigenomic circuitry, we've been involved in several consortia, including ENCODE and the Roadmap Epigenomics. And we basically integrated data that was generated in this huge collaboration with multi-institutions across the genome, across the world, not just across the US, that basically allows us to now paint across the genome-wide locations of all of these histone modifications, DNA accessibility, DNA methylation, and gene expression in both adult and embryonic tissues, multiple brain regions, and multiple primary cells and cell types. What this paints is a complex picture of the epigenome and the dynamic epigenome across tissues, which allows us to now start mapping genetic variants that are scattered across the genome into the specific cell types and tissues where they enrich based on their overlap with epigenomic annotations of those features and specifically enhancer regions. This gives us a map of hundreds of cell types and dozens of traits, where for every one of those, we can postulate that height acts predominantly in embryonic stem cells, immune traits in T cells and B cells, fasting glucose-related traits like type 2 diabetes, specifically in the pancreatic islets, cholesterol in liver, and then inflammatory bowel disease, both in inflammatory tissues and in digestive tissues. And interestingly, what we found is that Alzheimer's does not enrich in brain samples, but instead in this very surprising sample from monocytes, which are basically signatures of both the circulating macrophages in our blood as well as the brain resident microglia. And indeed, when we go back into those brain samples and we sort neurons versus microglia versus oligodendrocytes, we see that the lack of enrichment that we had seen in the neurons in the brain basically came from the fact that neurons do not contain genetic variants associated with Alzheimer's. Instead, those variants appear to act primarily in microglia. And that has now pushed us to a large program that basically tries to associate the cell type specific components of each of these contributors to basically understand how genetic variants are ultimately acting on disease. We're looking at both case and control individuals to understand what are the differences between them that we observe at each stage of the disease progression. And of course, when you have these intermediate phenotypes, you have to understand also how those are changing from disease itself and how the environment is affecting both. So we basically are building these statistical and computational models that allow us to infer, number one, the effect of genetics on the epigenome and on the transcriptome, the relationship between these transcriptional and epigenomic changes with phenotype, and ultimately combining them to basically understand truly causality of the disease. Here's one example of the first link between genetic variation and disease. And what we're finding is that there are tens of thousands of genetic variants that affect methylation in pretty substantial ways. So if I know the genotype for those individuals at conception, I can predict the epigenome of these individuals at 93 years of age in an inaccessible tissue. This is a big deal because I can now start inferring the predicted methylation using this genetic effect at thousands of loci across the genome, and then start relating this predicted methylation with disease itself in much larger cohorts of individuals. Doing this allows us to now paint the path of causality and discover new loci that allow us to predict both the directionality of effect and the causal variant and the cell type of action and the heritability captured in these genetic loci across the genome, predicting a series of both protective and risk-increasing genes in disease. But all of that is at the bulk level. To go to the single cell level, we've basically now started profiling more than 7.5 million cells across 1,500 postmortem brain samples, and of course, hundreds of other non-brain samples across Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's, psychosis in AD, schizophrenia, bipolar, Down syndrome, autism, depression, suicide, PTSD, and aging. In each case, we're profiling one or multiple regions, and in each case, we're profiling RNA, as well as very frequently single-cell DNA accessibility, capturing all of the major cell types. In the case of Alzheimer's, what this showed us is that genetic variants that are underlying Alzheimer's disease are in fact acting not just, of course, in the microglia, but that we observe changes associated with disease pathology and with cognition in all of the major cell types. Across a range of disease progression from non-AD to mild AD to severe AD, we're able to now start observing how the changing transcriptional programs for each of those cells is in fact associated with both genetic contributors and disease contributors. What this shows us is that every major cell type shows substantial changes between AD and non-AD individuals. You basically see that excitatory neurons subcluster into groups that are associated with pathology or no pathology, inhibitory neurons, astrocytes, oligodendrocytes, microglia. All of the major cell types are in fact showing this association. The other big surprise that we had is that if you look at male versus female individuals, the female individuals show a huge abundance of those disease-associated cells consistently across multiple of these cell types, indicating a higher transcriptional burden at the molecular level for female individuals. We find that changes early in the disease are associated with extremely cell type specific signatures, but changes later in the disease are much more common across cell types and very often related to stress response. We're finding that there are 3,000 differences between male and female individuals in terms of differentially expressed genes between them prior to AD, but with AD, this number doubles to 6,000 differentially expressed genes between male and female individuals, indicating sex-specific approaches are really needed to understand how to tackle Alzheimer's. And in our case, what we found is that myelination pathways are turned on by men in response to the disease, but women fail to turn those on, resulting in fact in much more white matter loss in female individuals. We're also able to now go into the single cell level at the epigenomic level, as we heard in the previous talk, and we're able to now start linking together known coding variants to their target genes at the single cell level based on correlations and co-clustering between RNA activity and single cell accessibility patterns between matching cells across these experiments. We're able to find coordinated changes associated with Alzheimer's in genes that are showing changes both in the RNA on the x-axis and in the DNA accessibility on the y-axis. Across all of the major cell types, we're seeing these coordinated changes between RNA and DNA accessibility, enabling us to now link the two. We're also studying how pathology and genetics are in fact both contributing to disease, cognition, and gene expression changes, by looking at ApoE2, ApoE3, and ApoE4 individuals across this gradient of homozygosity and heterozygosity, and how these are affecting each of the major cell types. And what we're finding is that these ApoE genotype-driven changes are in fact affecting very different cells in very different ways. In microglia, they're increasing stress, cell death, physical transport, immune signaling, and they're decreasing immune functions. In astrocytes, they're increasing proliferation, but they're decreasing transport. In oligodendrocytes, they're increasing cell adhesion and decreasing myelination. So there's a lot of cell type specific changes. We're also exploiting the sequencing-based nature of these assays to systematically infer the mutations that are harbored by each of those cells in order to recognize somatic variants that are only affecting a subset of cells and very often a very small number of cells. And what we're finding is that the somatic variants are accumulating in these neurons much more when these neurons are in fact showing signatures of disease, and that they are much more frequent in glial cells that are still dividing versus in neurons that are post-mitotic. We're finding that these differences in mutational burden are actually different between amyloid level and between sex, and that they correlate with aging, and that they're converging in specific pathways in each of the cell types. So if you look across more than a dozen individuals, both in AD and non-AD, you're finding a large number of genes that are showing increased mutational burden across multiple individuals. And those mutations can be mapped within the specific transcriptional context of each of those cells, because we're using RNA and DNA mutations jointly. And we can see how these are in fact converging on a large number of energy pathways in neuronal pathways. Since we've scaled this to thousands of individuals, we've basically multiplexed the profiling. We are looking at 1,500 postmortem samples. We're using that to predict QTLs and to infer changes across multiple regions of the disease. We're finding that the neurons in cortical versus subcortical areas have completely different transcriptional signatures, and each area of the brain is in fact showing a slightly different type of neuronal expression patterns, enabling us to now start recognizing the localization patterns simply from the expression signatures of these genes. So at the time of of spatial transcriptomics, we are able to now start looking at the distinct signatures of dentate gyrus of CA area 1, 2, and 3, and 4 in the hippocampus versus subiculum. And what we're finding is that our single cell non-spatial transcriptomics expression patterns are in fact capturing spatial information and enabling us to in fact distinguish multiple spatial sub-areas of the subiculum of the dentate gyrus of CA1 to CA4, enabling us to now start mapping the specific regions of the brain that are most associated with disease and the specific genes that are changing in each of those subclasses of regions. If you look at frontotemporal dementia and ALS, we've been able to look at, again, hundreds of thousands of cells across dozens of individuals and basically recognize a large number of pathways that are altered in each of these disorders that are associated with endocytosis, with apoptosis, with regulation, signaling, differentiation, and so on and so forth, and the specific genes in each of those pathways that are going either up or down in the context of disease. With schizophrenia, we're finding that the cell types involved are very different from Alzheimer's. They're primarily inhibitory and excitatory neurons, and those correlate very strongly with the genetic variants discovered from the PGC and other consortia. And we're again inferring the pathways underlying this, including a lot of neuronal pathways in each of those, as well as early developmental pathways enabling us to connect the two. And we're predicting master regulators of these processes that are acting both early in development and in later sort of axon and synaptic functions. In Huntington's disease, we're basically seeing that these indirect pathways, spinal projection neurons, are in fact merging with these direct pathways, spinal projection neurons in the striatum. And you're seeing that they're losing their identity in the context of Huntington's disease, consistent with a hypothesis that was actually raised quite a while ago. Combining these data with genetic information allows us to now start predicting the specific cell types where each of those genetic variants is acting in the context of disease, and sort of carry out these mediation prediction in all of the major cell types simultaneously, and then infer genetic variants that are mediating both the disease effect as well as cell type proportion differences between individuals. So we're able to now go back into individual disease loci and start fine mapping these genetic variants. And what we're finding is that they're very often localizing in microglial specific enhancers in the context of Alzheimer's, but in neuronal enhancers in schizophrenia and other cognitive traits. And we're also finding interactions between genetics and environment where specific genetic variants exert their effect only in the context of AD or non-AD. Going beyond individual phenotypes, we're able to dissect neuritic plaques versus neurofibrillary tangles versus neuroinflammation in the context of Alzheimer's, and then find the genomic region whose methylation level correlates with each of these pathological signatures. And these regions are in fact revealing very distinct pathways underlying each of these phenotypes. Expanding beyond Alzheimer's to all of the clinical records for each individual, we're now able to study the disease comorbidity patterns, identify multimodal clusters of pathology across clinical records, across doctor notes and prescriptions and medications and lab tests to basically recognize individuals in patient subgroups that are associated with each of these data modalities across multiple signatures, and then use that to now start cleaning up the clinical record, but also understanding the correlations between the genetic variants of these individuals that fall within these clusters and the expression patterns of those individuals as predicted from the GTEx project, the Genotype Tissue Expression Project, allows us now to predict what are the cell types and tissues where these genetic variants lead to gene expression changes, and then start associating these clinical phenotypes with genetic variants and transcriptional perturbations in a cell type specific way. Across multiple tissues, we're able to now look at the convergence of these changes and the communication that happens across tissues. Going back to obesity, we can see how exercise in obese or lean individuals or obese or lean animals is in fact manifesting in multi-tissue effects, where the exercise muscle is in fact leading to immune signaling across other tissues. We're able to actually predict the specific molecules that are secreted by these and how these are actually leading to both brain changes, muscle changes, heart changes, and blood changes across these different disorders. And we're finding a global role for immune cells in leading and mediating these changes. Looking at these tissue interactions, we're also able to look at blood methylation and cell-free DNA methylation as a biomarker of disease progression. And in the case of Alzheimer's, we're finding that the epigenomic alterations in the blood are actually quite predictive of Alzheimer's disease status in the brain of those individuals. We're able to look at biomarkers of aging by combining these transcriptional measurements and then use delta age as a phenotype and start asking how is aging and ApoE4 that I spoke about earlier converging onto Alzheimer's, and what is the interplay between the two. And we're also able to look at this convergence across a large number of non-coding regions that are targeting common target genes. In this particular example, in the case of cancer, we're able to look at the regulatory plexus of each of these genes and actually experimentally validate these interchromosomal convergence patterns through these interactions, and also monitor the progression of the disease, in this particular case cancer, across multiple time points and across multiple data modalities, looking at both genetic and epigenomic and transcriptional alterations. In this particular case, we're finding convergent events across the multiple lineages of the tumor that are coexisting in the same patient across multiple years of treatment, and how these are in fact evolving. This leads to a large number of predicted modulators of disease in each of these studies and the specific genes through which these are acting. And we're able to now start testing those systematically in high throughput. In collaboration with Kevin Egan, we're basically looking at many of these cellular phenotypes using sort of multi-well plates. In collaboration with Ligo Itai, we're building these programmable constructs to modulate both the genes, the promoters, and the enhancers, as well as individual SNPs. in iPSCs, which are then differentiated into neurons, microglia, astrocytes, oligodendrocytes. We're able to do this in 384 well plates to basically start carrying out these measurements systematically, again in collaboration with Kevin Egan's lab. And we've also developed a large number of assays, such as Hydra, that allows us to now measure the impact of these genetic variants across tens of thousands of loci in a single experiment, by basically capturing DNA accessibility, using those to put them in reporter plasmids, and then tasting the effect of these genetic variants on the ability to drive enhancer modulations. And in collaboration with Andreas Fenning, a former postdoc in my lab who's now a professor at TMU, we're basically now able to insert tens of thousands of these genetic variants simultaneously into constructs that are then inserted into the in vivo brains of mice, and then observe post-mortem what was the impact of these genetic alterations between control mice and disease model mice in a G by E interaction to basically understand the effect of each of those variants on genetic regulation of enhancers, as well as transcription alterations, as well as the physiological state of these animals. So to recap, we're basically building these reference epigenomes to predict disease-relevant tissues, regulators, and pathways. We're combining genetic, epigenetic, and transcriptional variation in the context of disease to understand the mediators, the causal genes, tissues, and the directionality within both known loci and new loci. We're dissecting this at single-cell resolution for both DNA accessibility and RNA expression, systematically across more than 1,500 samples, and multiple brain regions and multiple disorders. We're able to look at cell type specific genetic effects and QTLs acting at the single cell level across, again, thousands of individuals by combining both the single cell profiles and the convolution of bulk profiles. And then we're able to look at the individual subphenotypes of disease to dissect those signatures across the multiple tissues in which they're acting, and then understand the convergence patterns of those across multiple tissues, and to dissect these effects systematically using high throughput perturbation. What I'm describing is the work of many outstanding postdocs. Many of those have actually gone off to start their own research groups at Harvard, Columbia, CMU, Stanford, UCLA, and so on and so forth. So we're looking for postdocs. Please come join us. We're looking for both experimental and computational folks to join our group. And again, huge collaborations with both experimental and computational scientists. I'll stop there and I'll be happy to take any questions.
Thank you, Manolis. That was a really amazing talk covering a lot of ground. Just to give 10 people to put questions in the Q&A, I'm curious to ask you it seems that some of the resources that you guys are generating and your analysis would also allow you to look at pleiotropic variants that might have effects in multiple tissues. I was just curious if you could comment on that.
So absolutely. So we now have a lot of work looking at the pleiotropy of these genetic variants. We're basically able to build models for how is each of these variants acting. And here's some examples where we're basically seeing both, multiple examples of these type of association. We're able to take individual loci and map them to target genes when they're perturbing a single enhancer, or when they're scattered across multiple enhancers acting in the same tissue, or in some cases, when they appear to be acting across multiple regions, enhancers, and promoters of the same target gene, or when they appear to be acting across multiple tissues. So in this particular case, if you look at, for example, coronary artery disease, you see that genetic variants appear to enrich both in liver and coronary artery disease, and coronary artery itself as a tissue. And then you're seeing that different loci appear to be associated with different subsets. But there are also some loci that appear to be acting both in the liver and in coronary artery simultaneously. So in this particular case, pleiotropy is in fact acting across multiple enhancers in multiple tissues through multiple nearby genes, even through a single locus. So absolutely, we're seeing that pleiotropy is very much the rule rather than the exception. And it is pervasive at the level of convergence. And we're building all these convergence models, looking at, in fact, converging onto common enhancers, how multiple enhancers are converging into common genes, how multiple genes converge into common pathways, and how multiple regions or cell types are in fact converging. So I think that's going to be very much the model for understanding pleiotropy. Yes,
Okay.
every single one of these loci might act in multiple ways, but these are in fact ultimately converging onto common pathways.
Thank you. There's a few questions in the Q&A that I can read for you. So Sarah Marcy is asking whether you see differences in oligodendrocyte signatures across brain regions, specifically between midbrain and neocortex.
So not as much. Basically, if you look at the multiple analysis, so this is the cool part about sort of presenting on my laptop, that I can actually go directly into the corresponding slides. But for the multi-region analysis, if you look at oligodendrocytes, you basically see that they're largely preserved. The mammillary body is very different from the other ones, but they're, preserved across different brain regions. So this is very much in contrast with the neurons that are dramatically different between regions. So basically, these are the neurons across regions, and they don't agree much. And if you look at microglia or astrocytes or oligodendrocytes, you see a lot more agreement.
Thank you. There's another question from Emmanuel Levir. Amazing talk. I wonder whether Manolis would comment on the fact that brain disorders, unlike the other diseases he studies, can only be studied at endpoint. What are the consequences of this on data integration?
This is a great question, Manuel. Thank you very much. Because being in one of our grants for studying human, they say, oh, why bother studying human? You can only study it at the end stage. And I agree with that. I mean, of course, there's advantages to mice where you can sort of look across the disease progression. But what we're doing in the context of human is actually pseudotime modeling. So basically what we're looking at is how for pseudotime, we're looking at how, for example, every one of those individuals, we can basically start predicting the disease progression patterns transcriptionally, build predictive models for every one of those individuals, and then apply these models to the cells from each of the individuals, and then start sorting the cells in a sort of pseudo-temporal ordering across individuals. We can also sort individuals along the disease progression that they're going through. And start asking in pseudotime, not in real time, obviously, pseudotime across different individuals, sorted along disease progression. What are the regions that are changing? What are the genes that are changing? What are the cell types that are changing along that disease progression? So that's what we're doing now. We're basically building these pseudotime models using the multi-region analysis and using our 1,500 individuals to basically start looking at how disease pathology progresses in a temporal fashion. So again, great question.
Okay. Thank you very much. There is another question from Chris Bell, but we're apparently having some issues with the sound. So I'd encourage you to take the question onto the Slack channel.
Can I try to answer briefly to Chris?
Apparently they have issues hearing you. I'm not quite sure what the problem is. So the question is, are the somatic mutations you mentioned enriched within specific genetic motifs and or regions?
Yeah, so the answer is absolutely. It's a fantastic question, and indeed they are. So if you look at the genetic variants associated, the somatic variants associated with Alzheimer's disease, what you're seeing is that in excitatory neurons, these enrich, this is the enrichment for each of the pathways, these enriching in a lot of these neuronal energy pathways. The oligodendrocyte basically see that they enrich in a lot of these sort of biosynthesis pathways. So indeed, different cell types, they enrich in different sets of pathways. So basically vesicle transferrin, Golgi, intermediate filament, as well as many of these neuronal pathways of energy production are strongly enriched.
Okay. Thank you very much. So I think in the interest of time for the next session, so that people can have a bit of a break, I'll wrap up here. Thank you very much to all of our speakers and to Manolis for an excellent talk to wrap up the session. So as you know, for any of you that are that might not be aware, there is a different Zoom link for the next session. So either go into the program or Slack to join us later. Have a great break, everyone, and thanks again to our speakers for this session.
Thanks, everyone. Bye-bye.